I work a lot with livestock folks that I don't think they really understand managing nitrogen, because if you can't manage nitrogen, you have a hard time managing protein synthesis, and if you can't manage protein synthesis, then you start having problems with livestock health and productivity. You also start having problems with plant diseases. This report came out in the US just very recently, so we're in the same boat you guys are. This was a collaboration between NASA and US Department of Energy, and we're talking about the fact that whether they're cycles or whether they're exacerbated cycles that are influenced by man-made negligence and abuse, the point is, this is where we're at. And so the real question is, if we're wanting to get natural systems to create insulation against these extremities and create buffering systems against these extremities, we got to start understanding how nature works. I'm a member of the Pennsylvania Sustainable Agricultural Organization. It's called PASA, P-A-S-A. And if I had my druthers, I would change the name from P-A, Pennsylvania Association for Perennial Agriculture, I think is going to be more appropriate, but I can't do that right now. It's a 22-year-old organization, but I think sustainable, whatever that means, I think is passé. I think words like resilient to anti-fragile are now going to have to be looked at if we're really stressing the emphasis on buffering systems that can insulate us against floods and droughts. And so some of the work that came out recently that I found intriguing is understanding that natural systems already have innate buffering systems already built in. And this was about transplants. This is the hort folks, or even the crop folks here. But they found out that if transplants were not watered when they were put out, when they were exposed to stress a few days later, that they ended up having much more resistance against wilt. I don't know if any people ever heard of IMOs, or indigenous microorganisms, that came out of Korea. And that's one of the things that they're always talking about doing, is don't water your transplants for up to a week. You'll have a lot more resistance and resilience. And it's because plants have a molecular memory. And as I talked about in the earlier talk, that memory is probably in water, because water has a memory. This is of real interest to me, because we usually talk about the soil microbiome, and the microbiotic communities being fungi, bacteria, arthropods and such. We never talk about the other guys, the viruses. And the reason why we don't talk about them, we usually associate viruses as being those guys that create crop losses or diseases and animal diseases and death. It's the viruses that actually are some of the best buffering mechanisms within the microbiotic community. The microbiome community is very, very, in a healthy sense, a very, very diverse community. And we've actually found out that 7,000 plants that they've examined for viruses, most of them were actually very, very beneficial, because what do? They move around information. And the way they move around information is with this Dr. Seuss diagram that I scribbled out before I came here. And what we're showing here is basically bacteriums. This is the bacterium here with the DNA in the middle. And the phage DNA enters the host cell. This is the virus. They call them a bacteriophage, or prokaryotes. So these viruses come in here, and they basically put their DNA in here, and eventually the host DNA is digested, and eventually a new DNA is created, creating these baby viruses which explode out of the lyse of the bacteria cell, creating this whole cycle again. Or another diversion or detour is the phage DNA integrates into a bacterial chromosome, and they actually create new bacterium. Completely different bacterium. What's the point of all this? Well, the point of all this is that information gets moved around from bacteria to bacteria, from different plant kingdoms, or I should say microbial kingdoms, like fungi to fungi, fungi to bacteria, protozoa to bacteria. So what we're saying is that viruses, or the bacteriophages, and you can see what they look like. They actually look like an Apollo device. They're responsible for controlling these bacterial populations, because they actually consume them. And in the process of consuming them, the DNA is actually distributed throughout the entire microbial community. So genetics actually is moved around. Bacterial genetics is actually moved around thanks to the bacteriophages, or viruses. It's especially effective in the presence when you have montmorillonite clays and humic materials. And it's especially increased in its efficacy when you have a high amount of plant root exudates. Humic acids which come from carbon-based raw materials like compost or plant exudates, in combination with all of the variety of the dispersal of the photosynthetic compounds that are made above the ground surface, create this wonderful opportunity for communication to occur. And communication is all about what? Well, your immune system only can work well when you have a very good communication system along with. So this is plant and soil immunity. It's communication, moving information around through viruses. They're involved in the transfer of DNA from other bacteria species, DNA from other species organisms, transfer of DNA from other kingdoms like higher plants. So in other words, we're saying that the DNA that are in higher plants actually are moved around through the microbiotic community thanks to the bacteriophages. And the real concern about GMOs now is, does this actually happen? Transferring DNA from transplasmatic GMO plants. Why wouldn't it? That's the real question. That's the $64 question. And nobody's really looking at this from their side of the fence. But if bacteriophages are that incredibly capable of moving DNA particulates around through the microbiotic community and into the plant community, ultimately into the animal community, I mean, we're all one. Why is it that we can think that genetically modified interferences would not be affected by bacteriophage transference? Pesticides. How do pesticides increase pests? Well, basically what we have is we have an inhibition. One of the things I want to talk about is, again, protein synthesis as it relates to nitrogen, because, again, protein is made up predominantly of the element nitrogen, but certainly not only nitrogen, which is what we typically think of when we're simplistically trying to overcome a resistance to understanding the microbiotic community with strictly an NPA mentality. The metabolic problem is we can't create finished proteins. So if we're not creating complete proteins, then we have funny protein. In animal forage rations, we call that non-protein nitrogen. And in horticultural crops, we call that incomplete protein. And the bottom line to incomplete protein, funny protein, or non-protein nitrogen is the fact that we now have a fantastic substrate, a protein offering for primitive organisms like fungi, bacteria, and insects. Being primitive organisms, they have primitive digestive systems, and so they love incomplete proteins because complete proteins are very, very difficult for them to enzymatically consume. They don't have sophisticated digestive systems, therefore they don't have the array of enzymatic systems to consume these complete proteins. So therefore, by having a lot of funny protein in the plant tissue, you end up with an appetite banquet for pests. When that incomplete protein called NPN ends up in an animal, including people, we end up creating substrates in the body that invite diseases because we create high levels of what we call blood urea nitrogen, BUN. Blood urea nitrogen is a substrate to grow pathogens. Blood urea nitrogen also robs oxygen out of the system. Blood urea nitrogen also attacks the liver and the kidneys so that they can remove it. And when the liver and the kidneys are taxed to remove blood urea nitrogen, what happens to the animal? It suffers an immune collapse, or it can't reproduce, or it can't put milk in the bucket, or it can't put meat or muscle on the frame. Because an animal can only detoxify to save its life before it does anything else. A dead animal doesn't reproduce. A dead animal doesn't put milk in the bucket. So these metabolic problems that we have in plants and animals are very similar. It's related to not managing nitrogen very well. Factors affecting diseases and the availability. This happens to be just one trace mineral that, how many people heard of Dr. Don Huber, the guy who's talking a lot about? Well, he's a plant pathologist and a plant geneticist, and he's retired now from Purdue. I actually have about 5 or 6 of his plant nutrition textbooks from college, not when I went to college, but from his college days. He's authored like 50 books on the relationship between mineral nutrition and plant growth and resistance to diseases. This just happens to be manganese related to take-all disease in wheat, showing that, by putting in too much nitrogen in the system, you end up creating the wrong form of manganese, or you drop the levels of manganese in the sense that you start increasing take-all diseases. So imagine how many minerals are involved. I mean, I think we heard this morning that we're talking about 70 some minerals that are probably potentially important to optimal plant growth and development. So nitrification is something that all agronomists understand. It's talked about in Agronomy 101 and Soil Classes 101. So we have different organisms like Nitrosomonas and Nitrobacter that actually convert either ammonium to nitrate, or you got these other guys in here called Archaea, which are the primitive pre-bacteria bacteria that actually showed up before bacteria showed up on the planet. And there's twice as many or more archaea than bacteria capable of oxidizing nitrogen. So what does that tell me? That tells me that you only get archaea when you have good humic content in the soil. I don't know how many people realize this, but here's another elephant standing in the living room, kind of like silica, which is one of the largest known mineral elements in the Earth's crust, is that there's about 30,000 tons of nitrogen gas hovering over every acre of land, and that's not a coincidence. So we're looking at nitrogen gas as the primary raw material to make protein, and that can only be done when you have microbiotic communities able to somehow take that gas and convert it into nitrate or ammonium. But plants grow best when you have a combination of nitrate and ammonium. And the reason for that, according to Don Schriefer, one of my teachers back in the 80s, was that there's less enzymatic requirements to convert ammonium into protein precursors than there is to convert nitrate into protein precursors. So we like a little bit of ammonium in the soup with the nitrate so the plants don't have to expend as much energy to synthesize complete protein. So the nitrate ends up in the cytoplasm of the roots and the shoots, and the ammonium ends up in the chloroplast or the leaves. Nitrate conversion, here's what I just said, requires significant enzymes and energy. There's 15 moles of ATP versus 5 moles of ATP to convert to ammonium. So that's why you won't see as much ammonium in the soil as you will nitrate, but a little bit of ammonium goes a long ways in being able to have that plant conserve energy to send those compounds, those energy compounds, to other important parts like filling fruit or putting sugar in the leaf tips. Obstacles to photosynthesis: too much nitrogen. Excess nitrogen actually creates an uptake of too much water. An uptake of too much water with nitrogen creates this funny protein phenomena. Funny protein phenomena creates the appetite for insects, diseases, and sick animals as a consequence. Deficiency of proteosynthesis. Proteosynthesis versus proteolysis. Proteosynthesis is making complete protein. Remember, there's about 22 amino acids, and that's it, only 22. And yet, we're talking about building the potential of 50,000 different proteins with just the various combinations of the 22 amino acids. And that takes enzymes to do that, and enzymes depend on macro and micronutrients. They also depend on cofactors like vitamins in the process to make these finished products. Pesticide applications interfere with proteosynthesis. Mineral ion shortages interfere with it. Dry conditions or compaction, I talked about compaction this morning, which infects uptake of water and minerals. Toxic irrigation water. Out west, western United States, we have a lot of recycled municipal wastewater that's loaded with excessive amounts of carbonates and bicarbonates, also very high levels of sodium, but that's the only water they have because they get 2 inches of actual rainfall a year on some of these irrigated croplands and golf courses in Arizona and Nevada. So you're really stuck with a situation where the water is so detrimental. To not have water means no crop. To have salty irrigation water means you're going to have basically diseases and insects because there's no way that plant can use really good water or bad water that way and end up with a finished product that you're trying to have in a healthy crop. Recycling of mobile nutrients to younger tissues. As the older tissues senesce or they die, those amino acids are recycled down to the younger tissue. So what does that mean? Well, if you don't have finished proteins, those leaves that haven't finished making protein prematurely die, and so there's constantly a recycling of that nitrogen. Chlorophyll is an example of nitrogen. In the Northeast, we have autumn, and autumn is when the leaves change color. And really, they don't really change color. What happens is they lose the green color, which is chlorophyll, because chlorophyll is rich in nitrogen, and the deciduous trees migrate or mobilize that chlorophyll into the root for next year. Through the wintertime, it stays dormant. and then it resurrects that nitrogen back in the spring. So what you see under that chlorophyll mask is this rainbow of orange and yellow and red, and these are all the carotenoids or carotenes that you see under the green pigment. Plant proteolysis, so high nitrogen fertilizers. This is the problem I'm talking about. Excessive amounts of free amino acids in the plant tissue. Okay, so protein is a mixture of bricks. The bricks are amino acids that hook chains together called peptides. The peptides hook together and make basically blocks, and blocks hook together and make this wall. That's the complete protein process. If you don't have that process finished because of enzymatic deficiencies and other reasons of interference because of pesticide interference, shutting down the enzyme systems, or too much nitrogen, you end up what they call free amino acids. In other words, they're not linked together. And free amino acids are, in effect, a buffet for diseases and insects. That's one of the reasons why trace minerals like boron are so effective, oftentimes, in arresting the development of insects and diseases, because boron and manganese and copper and zinc are catalysts in producing these enzymes that allow these finished protein compounds to be accomplished. So what we have is, in effect, the xylem, where things are taken out of the soil here. This is going up, but it's actually laying down. And so we have nitrate and ammonium, and we have amino acid metabolism forming these free amino acids. Carbohydrates are produced by photosynthesis. Carbohydrates plus amino acids make true protein instead of funny protein. And then half of this is dumped down into the root systems. That's what we call the nectar of the roots of the rhizosphere. This is, in effect, nurturing the livestock of the soil with these compounds that they need, along with all the aromatic compounds. Things like alkaloids that actually inhibit the proliferation of nematodes, things like terpenoids, which inhibit the growth of fungal diseases that attack root systems. Plant immunity depends on the following: cross-linking proteins. Cross-linking, in other words, coupling, hooking up instead of having free amino acids. Cell deaths. Surround fungus releases acids to contain the pathogen. So that takes a lot of energy. When a plant's being attacked by a fungus, there's a tremendous amount of energy focused just on where the damaged tissue is, which takes away from what? Production. It also takes away from the quality of the fruit if you're growing fruit or nuts. Surrounding cells produce phytoalexins. What are those? That's and I'll talk about this Saturday, I guess. Phytoalexins are the 911 responders. Phytoalexins happen within a very short period of time. Within 48 hours of a plant being smitten or bitten by a pathogen, the entire phytochemistry of that plant can actually come to the aid of that damaged area so that the disease does not progress and there isn't a crop failure, provided that there's enough nutrition, provided that the protein is not funny protein. If you have an abundance of this funny protein, you can't produce the phytoalexins, these 911 responders. And these are some of the things that are in these phytoalexins, or these 911 responders. So we have things like phenols, hydrocyanic acid, that's cyanide. Which plants are high naturally in cyanides? Brassicas, right? They're called isothiocyanates. It's one of the reasons why they're so good against cancer, because cyanide is actually very good for preventing cancer. You know, there are lethal doses of cyanide, and there's therapeutic doses of cyanide. Anybody ever hear of Laetrile? Laetrile from apricot pits. Got vilified years ago. The active ingredient in Laetrile or apricot pits is another form of cyanide. And cyanide compounds in these plant forms are incredibly effective at detoxifying the liver. So when you have cancer, or you have some other kind of chronic illness that has an association with liver contamination, you want to eat lots of cruciferous vegetables, particularly if they're fermented, because they cause a conjugation of the toxin. In other words, it binds it and it helps you eliminate it through the gallbladder. And plants do the same thing. These are conjugators, right? They actually pick up toxins. Glutathione is another conjugator. Plants and animals and people work on very, very similar principles in terms of detoxification. Organic acids like citric acid, acetic acid, which is also called vinegar, one of the best panaceas for human and beast. Also a great additive for your foliar sprays. Anybody ever use acetic acid in your foliar sprays? Anybody use it? Yes? No? Try it sometime. Yes, you use acetic acid and vinegar in your foliar sprays?
Mixed with malt.
Mix it with what? Lye. Oh, lime. Oh, to make the lime more available, right? Okay. So amino acids like glycine and methionine, phenylalanine, these are all precursors to make things like glutathione. And then also foliar sprays, you can manipulate, it's called induced systemic resistance. Anybody hear of induced systemic resistance? You know, SARs are acquired systemic resistance. That's when plants cause this immune response to occur where phytoalexins, the 911 responders show up. You can actually trick plants into doing this with things like vitamin C or salicylic acid, which is aspirin. And if you're organic, I don't think you can use a synthetic salicylic acid, but you can make your own by taking willows, chopping them up, putting them in water, letting them leach into the water, and using the salicylic acid from the leaching of the willow branches and leaves. Phenols and mycorrhizae. Okay, so what's this saying? This says that the mycorrhizal fungus, that is a beneficial symbiont, that is a partner with the root, of which 90% of the world's plants are now mycorrhizal dependent, either ectomycorrhizal, which live on the outside of the root, or endomycorrhizal, which parasitize the inside of the root. The mycorrhizal fungus is incredibly important because of a couple of big things. Number one, it produces a lot of a plant glue exudate called glomalin. And glomalin doesn't break down, so it builds up organic matter. Up to 40% of the soil's organic matter is from glomalin. It also insulates the plant's roots so that they're not susceptible to diseases and insects, salt damage and such. And these mycorrhizal also produce phenols, phenolic acid. Anybody ever hear of phenolic acid or phenols? It's used as a preservative because it kills bacteria. And there's a whole family of phenols. They're part of the flavonoid family. But phenols are secreted by these mycorrhizal fungus. Unfortunately, you can't see it, but those dark shaded areas there are actually phenol deposits coming out of the mycorrhizal fungus. And what this shows you is total phenols of mycorrhizal infections and on groundnut roots. And what this is showing you here is the mycorrhizal inoculated, the non-mycorrhizal inoculated. And these are different phenols. So here's total phenol, here's a particular kind of phenol, and this is the fact that these are mycorrhizal infected roots and none of these are infected. So you look at the two columns here and what do you see? Huge increases in the phenol expression out of the mycorrhizal fungus on those roots when they're inoculated. So if you're growing hort crops, I would always inoculate your hort crops with mycorrhizal inoculants. If you've got a perennial polyculture, you're already there. You just don't kill them. The best way to get rid of mycorrhizal fungus is apply too much phosphorus. Number one detriment to mycorrhizal getting rid of them is apply too much phosphorus. The worst kinds are the most soluble phosphates. So liquid manure, superphosphate. You guys, if you're organic, are not using that. Some guys are biological. You might be using some MAP or DAP or superphosphate. That's soluble. I'm not talking about adequate amounts, but abundant amounts. When you start getting the soil profiles up high, we're finding out as the mycorrhizal levels start to drop off. And the reason for that is because the primary job of mycorrhizae is to produce phosphatase enzyme. Phosphatase enzyme fractures the bond between phosphorus and iron, phosphorus and aluminum, phosphorus and calcium, and phosphorus and magnesium. So its job is to release phosphorus, and the reason for that is because the energy currency of all plants and animals and people is a molecule called ATP, adenosine triphosphate, right? So mycorrhizae's job is ultimately to create energy currency. And so that's the number one way to get rid of it. The second way to get rid of it is a lot of tillage, no cover crops. The third way to get rid of it is fungicides and herbicides. You know, so those are the ways you drive mycorrhizae out. Funny protein versus true protein. So this is Albrecht's work along with some colleagues, 1950. All this is showing you that sulfur plays a big role in creating complete protein. They didn't call it funny protein back then. You need sulfur. In the forage analysis labs, we look at least 10% of the nitrogen content having to be sulfur. Weeds was talked about this morning. You find out that those weeds that are really, really high in protein are also very, very rich in sulfur. And that's really important because what we find out in these high protein weeds and these high protein herbs, they don't create a lot of rumen ammonia, they don't create a lot of BUNs, blood urea nitrogen. Why? Because they're complete. And this is another biological value of alfalfa slide that shows you have to have sulfur in the equation. Most of the soils that I've looked at samples of are very, very low in sulfur. And that's not surprising, particularly on cropland, because sulfur, being an anion, it has a negative charge. It tends to leach away from the negatively charged clay colloid, and so it's a leachable element. If you have high organic matter soils, it tends to hold on to sulfur more efficiently. So there's lots of things that make up organic matter. Or living organisms are less than 5%, fresh residues 10%, decomposing or the active fraction is up to 1/2, and the stabilized or the humic fraction is up to 1/2. And these are all the subtypes of those types. So you have fats and waxes and lignins, tannins, all the pigments, the color of the rainbow, the sugars, organic acids, the aromatic compounds. And what this is showing us here is basically nematodes and protozoa, and why you need the food web in that hierarchy so that you can actually create these ratios of bacteria, which are high in protein, to protozoa, which are higher in carbon, to predatory nematodes, which are yet higher in carbon. And the reason why we do that is because as we create a bloom of protein, we create consequently a bloom of nitrogen. That's feed for the predators like the protozoa, and for the predatory nematodes to come in and harvest that high protein diet. And when they eventually expire, their higher carbon bodies leave higher carbon residues. So I'm going to spend some time on this guy because this is one of my favorite pets, earthworms. This is Lumbricus terrestris, and I talked about this morning, about how much earthworm castings you can get, basically in a cubic foot of 25, a reasonably attainable goal of earthworms. You can get about 30 tons of earthworm castings per acre per year. So Darwin was the guy who really got very, very infatuated with this, and everybody, basically either credits him or vilifies him for his evolutionary theories, but really a lot of what he wrote on and studied on was the earthworm, because this guy was an observer of what healthy soils were all about. And this was the title of the book. And in chapter 4, you read this book, it said Darwin placed stones on pastures and waited 29 years to grow 7 inches of castings, or 1/4 of an inch per year. That's all he did. Didn't do anything to it, right? The influence of earthworm activity on soil microbes. This is out of Wade Elmer's work up in 2009. He's an extension researcher at the University of Connecticut. And what they did is they basically took 2-liter plastic pots, they mixed them with soil peat mix, 10 grams of dolomite, and they fed the earthworms alfalfa 2 grams a week. They had a little bit of a fertilizer treatment. They had a manure, dehydrated cow manure treatment every 2 weeks, and they put 4 worms per pot, which is equivalent to 250 worms per square meter, or 25 per square foot, which is what my number is. Soil temperature was 19 C, and ended up with basically dry weight of the plant. This was on eggplants, on growth and diseases, severity of verticillium wilt in greenhouse studies. You can see the manure alone without earthworms. The manure earthworms over here showed you 5.5 recovered, fertilized with 20-10-10 fertilizer, none recovered, autoclaved and fertilizer, none uncovered. And what are we seeing? Dry plant weights? Asparagus? What are we seeing here? Root lesions. So fusarium colonies were downplayed on time to go into all of this stuff. Tomatoes, basically fruit weights went up with earthworms versus a control by about 250%. This was on microbial communities, pathogen-infested soils from the rhizosphere of asparagus, eggplants, and tomatoes grown in the greenhouses. Basically, what's in red is the earthworm treated groups, but what you see are the synonymads are much higher in the earthworm groups. These are beneficial bacteria that showed up primarily because they're coming out of the digestive system of the earthworms. The bacilli are better, the total bacteria are better, the manganese oxidizers, trichoderma, all of these things get better when earthworms are in the picture. I'm not going to spend much time on this, but this is, again, a cultivated field from 1942. And what you see are all these minerals: carbon, nitrogen, calcium, magnesium, exchangeable calcium, percent of total calcium. All of these wonderful numbers go up when earthworms are there in abundance. Soil averages 0.00056% ammonium. Worm castings average 0.018% ammonium, a 321% increase. British earthworms, now this is Darwin's work, add 10.59 tons of castings per acre per year, which is somewhere between 25,000 and 50,000, and that's where the 381 pounds of ammonium per acre per year come from. That's a lot of fertilizer. Comparative characteristics of earthworm casts in soils. 35 centimeter tall grass cylinders, 2 anisic earthworms, and what do you see a week later? This is from the Nature and Property of Soils, the textbook in colleges right now. You see that there's a tremendous number of distribution. If there's no earthworms, all of the nitrogen is distributed in the litter. When you have earthworms, a lot of it's in the soil, some of it's lost as gas, but it's not really lost because it's nitrogen gas that if you have nitrogen-fixing bacteria, recycle it again, right? This is in the earthworm itself, this is what's left in the litter, and this is what's in the plant. So what we're talking about here is if you don't have earthworms, most of it's ending up in the litter. What good is that if it doesn't get distributed through the whole soil profile? White Nile Valley, the Sudan, one of the most fertile places on the face of the earth. 6 months growing season produces 239,000 pounds of castings per acre. Estimated 12-month soil analysis at over 200 tons of castings per acre per year. That's amazing. Fed by the Blue Nile from Abyssinia at 9,000 feet altitude. The river is 17% silt, 9% organic matter, 8% minerals in the floodplain. No cultivations required. All the cultivations done by earthworms. I know an Egyptian fellow that lives in Pennsylvania, and he told me, he said the Sudan could feed all of the Middle East. The soil is that fertile. Could feed all of the Middle East. Of course, political issues are preventing that from happening. Topsoil generation without earthworms, 1 inch requires 500 to 1,000 years. With earthworms, 1 inch requires 5 years. 1 acre contains somewhere, depending on your soil, 1 to 2.5 million earthworms weighing 560 pounds to 5,000 pounds. Every 24 hours, body weight of the soil passes through earthworms, which is anywhere from 1/4 to 2.5 tons. Our average U.S. soils are somewhere between 250,000 to 2.5 million earthworms per acre. They're ecosystem engineers. They don't only churn the soil, but they also change the habitat of other plants. Slash and burn Peruvian rainforest studies that were done. Maize yields increased 23 to 60% in the first season with earthworm inoculation and mulch. New Zealand pastures. This is an interesting 5-year study that was done. This Lumbricus terrestris was actually deliberately inoculated on those pastures. What they ended up with, 25 worms inoculated in 10 by 10 meter grids. Soil productivity increased 70%, 20% more ryegrass. The breeding ewe flock doubled, an extra 4,000 pounds of wool, and up to 8 million worms per acre per pasture. That's an explosion of worms. But that made a huge difference, and that was done back in the 20s, up to the 1940s. So this is the Kingdom of the Earthworms. There's basically 2 orders, 23 families, and 7,000 species. I don't know how many species you have in Australia. These are the 3 basic groups. The epigeic species, and then the deep burrows, which I talk a lot about, this Lumbricus terrestris guy, the night crawler. You have them out here, night crawlers? Lumbricus terrestris? No? You don't have Lumbricus terrestris here, really?
Not surviving in the pastures.
Not surviving the pastures. Can survive New Zealand pastures, but not Australian pastures. Boy, I won't take that back to them. Okay, so a domestic worm bed propagation. 3,000 worms per cubic foot is 130 million worms per acre foot. 500 worms is a pound, it's 31 worms per ounce. Average is 4 inches long, 2,000 pounds of worms is 1 million. The goal, 10 worms per cubic foot at 30 inches depth. That's a very, very modest goal. We go for about 25 for that depth. All right, let's just go to Q&A, because I'm on it. Well, I mean, the research shows, I mean, Lumbricus terrestris came over the United States with the colonists, you know, and in the Connecticut River Valley, which goes up through Vermont and New Hampshire, that area was considered fairly infertile, even the alluvial bottom soils of the river valley, until Lumbricus terrestris. We have native earthworms there. Earthworms, where they're not desired, Lumbricus terrestris, is in the northern forests of like Minnesota, where you have wild orchids growing. And wild orchids like to colonize leaf mold. And earthworms turn leaves into soil, into humus. So the leaf mold disappears. And that primarily was from fishermen dumping their fishing worms after they got off the lake into the woods. And those worms migrated into the forest and ate the leaf mold. So there's this sign that says, take your fishing worms home with you. Because they don't want the leaf mold to be digested. But I'll tell you, that process of turning leaves into topsoil is a fast process, and on pastures or on cropland, it's astounding. So I'm surprised that you don't have Lumbricus terrestris here, because I'm sure, I can't believe it wouldn't do well here. I mean, I don't know why it wouldn't do well here. You might have them, you know.
Well, it's been here, but they say the survival rate's really poor. I guess maybe the conditions of the dry, etc. But on the coast here, Amynthus cordicus and Apparectidae.
Are the other two? And they're natives or are they imports?
All imports.
All imports.
Yeah, and we're looking for the deep burrow. We want a deep burrow to survive.
Yeah, and that's what Lumbricus is a deep burrower. And that's, I mean, even in the middle of a drought and where the soil's got cracks in it, you know, that wide, I mean, they just go down, all right? They just recede and they go dormant and they're back at it, you know, when the rains come back. I mean, you can't eradicate them. So I think somebody needs to bring them in there. It won't be me, because they wouldn't even let me bring radish sprouts through the customs. So, but I think somebody needs to try to get Lumbricus terrestris in here, because I think where you have, what is your average rainfall in this part of the country right here?
We just had 40 inches.
Yeah, that's, I'm saying average, not abnormal. Different A, average. What is it? 30 inches? 40 inches. Yeah, that's Pennsylvania. So I can't imagine them not surviving. But
Well, there's other Lumbricus that are around, not terrestrial. And I think that's been the thing surviving when we have
the flooding
well,
or the drought.
Well, they do. Well, they run out of oxygen in the wet and then
Yes.
dry out in the dry. Some of them say the resilience required haven't quite got that species covered, I don't think, for a lot of the Australian environment. But there are others that we're going to keep chasing the ones that can survive and burrow deeper than the ones that don't.
So the worm farmers that actually create worm teas and vermicast and all that, they're using what, red wigglers for that?
Yeah,
Yeah.
reds and tigers.
Right.
And they can't survive in the paddock.
No, they don't survive in a paddock either. Yeah, they're strictly for commercial purposes.
Yeah.
Is anybody raising red wigglers to feed birds, chickens? We have guys that are raising worm bins as a tremendous protein and fat source for chooks. And it's really, really effective, because they virtually cost nothing to raise. You don't have to buy feed for the worms. They live on residues of crop waste. It's amazing, you know. And the protein content is a very high quality protein, and the fat content is a very high fat content, so the energy's there, and it can cut the feed bills down tremendously on raising chooks. So you might want to raise worms if you're raising chicks over there, Chris.
And you feed the worms to chooks.
You feed the worms to the chooks. All right, and they'll eat nothing else if that's what you feed them. We get that all the time. And I live in a narrow floodplain, which floods seasonally, and you always see you know, drowned earthworms, but it never stops the production of the posterity. And so it's not like humans. Yeah, right. Yeah, no, they keep coming back. In fact, it's the worms that look like Lumbricus, and we call it the pot worm. They're a worm that tends to live in the rhizosphere and the root ball, and they tend to migrate laterally, and they get hit first with flooding because they're in shallower soil, which is much more prone to being flooded first. But you'll see a lot of those worms on the streets, or out on top of the driveways and the lanes, the farm lanes, or on the cow lanes. You'll see them. They're climbing as much as they can. But every year you see huge numbers of those. The biggest thing I see with earthworm death that concerns me is liquid manure, because liquid manure is loaded with ammonia, and ammonia is a very toxic compound for earthworms. And where you see the slurry wagons go and dump liquid manure, even on the organic farms, you see a huge swath of dead earthworms because of the ammonia content. And the indole scatol levels are so high in the liquid manures. Very toxic stuff. Yeah. And high in soluble phosphates, so it's toxic to the mycorrhizae too. No, they need something that's not finished. Compost is finished, so they don't need compost. They make compost. So basically, vegetation, you know, waste. Manure, raw manure. Raw manure, right. Vegetables, yep. Castings. Casting away, I should say. They make the castings. But, you know, a lot of people have them in their homes as their garbage disposal system. You have one? The worm drenches. Well, I'll tell you what we do out east. We use edible clay. And I think you probably have a few sources of edible clay in Australia. Well, it's a montmorillonite. It's the one clay that we use is what they call a hydrothermic or a geothermic montmorillonite. It's an old mud pot is what it was. That was a hot spring. So the minerals are fractured from the heat. And so the availability of the minerals is better. But some people use bentonites, calcium bentonite. Another thing that some folks are using is soluble kelp and soluble humic acids. And I've seen some remarkable things, and it actually works as a drench. There's another mixture of minerals that were put together, and there's a lot of these deposits all over the United States, and I'm sure you have your equivalent here in this continent. Humic acids mixed with montmorillonite is a fantastic way of creating a parasite-free zone, particularly with the animals that are on pasture and not constantly exposed to the same negative environments of confinement, where there's a buildup of cryptosporidia or coccidiosis or Homunculus, barber pole worms, and all these things that are cumulative, you know, in a confinement environment. So yeah, the clays and the humates, kelp mixed with that. There's one group out there puts this thing called NKS. It's kelp a third, sea salt or regular rock salt a third, and then a mixture of humate and montmorillonite a third, and let the animals just free choice it, and they don't ever have problems with worms. Or sometimes they'll throw in some sulfur if the flies are bad, and that seems to help with the internal parasites as well. I'm out of time. Thank you, guys.